And our presenter today is actually the facilitator of this whole course, and I couldn't do justice in introducing her, but Jane Brockmann is quickly becoming one of our fan favorites as a presenter. So we appreciate you being with us, and I'm going to leave it over to you.
Okay, so yes, it is working. All right, well, thank you very much, and thanks for everybody here and on Zoom. I'm really looking forward to this semester. All these wonderful colleagues of mine who are all talking about the things that fascinate them the most. So I think it'll be a really fun semester. Yes, I'm Jane Brockmann and the facilitator. And if I could have my PowerPoint. Yeah, so I taught animal behavior at the University of Florida for 35 years, and I always like to begin my course with this picture because it suggests to me that there have been people interested in animal behavior for a very long time. Yeah? Yeah. I haven't been in that cave, I've been in some others, so that's cool. Yeah, one of the things that's most fun about this, it's obvious that there's the artist portrayed two European rhinoceros of different sizes. The horns are different. The leg positions are right. Even the tail positions are right, as you see, one up and one down. So people have been fascinated by animal behavior for a long time. So I thought I would begin by just saying a few things about the field of animal behavior and then move into talking about some of the research I've done on horseshoe crabs. So animal behavior, in Europe it's called ethology, but here we have animal behavior society, and the focus is on what individual animals are doing, and it's a focus on the natural behavior of wild animals. There are a few animal behaviorists working on domestic animals, but most of us work on animals in the field, or we do sometimes bring them in for specific questions, bring them into the lab. But most of the work is informed by field work. Now, the first point I want to make is that there are really two very different kinds of questions that you can ask about behavior. We call them proximate and ultimate, or how and why questions about behavior. So the proximate determinants, or the proximate factors are the immediate factors affecting the behavior of the animal. Things like the cues that the animals are using, the hormones that are affecting their behavior, the environmental conditions, the immediate mechanisms of behavior. The ultimate or why questions about behavior, why explanations of behavior, deal with the function of the behavior. Why is the animal doing such a bizarre thing? Why are they doing these things? And that has to do with the selective advantage of the behavior, how the behavior that they're performing improves their individual reproductive success. But the important point about this is that you need both proximate and ultimate explanations to fully understand behavior. So there have been a lot of disputes. Oh, no, that's not the real explanation, it's this. And mostly those are disputes about proximate versus ultimate explanations. The second point I want to make is about variation in behavior. This is, behavior is measured very much in the same way as any other trait. Behavioral traits, we would measure the variation in a population, maybe in something like the frequency or the latency to chirp or to do something. The distributions don't always look as nice as this, I'll say, but the same idea of measuring the variation in the trait. In this case, the trait has been measured, the height of this class, 1910 class, has been measured in inches, by each inch. But of course, you could have just had the short, medium, and tall individuals, have them in groups. in categories. And there are statistics for handling categorical data that work very well, and we very often use those in behavioral research. Now, the reason I'm using this slide to make this point is that some of the variation in height, when you see this distribution, you will understand that some of the variation is due to genetic differences between individuals, and some of the variation is due to environmental differences. So by genetic differences, I mean that the trait is heritable. That is, the individuals at this end of the distribution, at the tall end of the distribution, have on average taller parents than individuals at the other end of the distribution. So the trait is in part heritable, but it is also strongly environmental. Some of the differences between individuals are due to environmental, maybe prenatal conditions. So this is true of virtually all traits, is that they are affected by both genes and environment and gene-environment interaction effects. And this is true also of behavioral traits. So we don't really, most of us don't really work very hard at, I mean, we just are assuming that there's heritability, which means that the trait can evolve, and we don't kind of worry too much about that. And that's important because when you're studying ultimate functions of behavior, the ultimate, the why questions of behavior, you have to assume that the animals the behavior can evolve. And the third point I wanted to make, sort of introductory point I want to make about behavior, is that it is very tricky to talk about behavior. And that is, of course, because you have to use words. And words have meaning. They have meaning other than what you necessarily mean to portray when you're assigning a name to something. So for example, we have assigned the word queen to the reproductive individual in a colony of honeybees. And that term really biased the way in which people studied honeybee hives for a very long time, because queens are not monarchs in any sense of the word. They do not behave in that way. And so we were biased. We were unable to kind of get away from our metaphor. We were unable to see what was really going on in that colony because of that bias that was just brought in because of the word that was chosen. Here's another little example. When you see a pair that is of birds living together, taking care of offspring together, sitting next to each other all the time, we assume that, we call that a pair bond, and we assume that it was, well, we understand about pair bonds. You know, there's caring and love and you know, affection and so forth. And that was the way we referred to this behavior for quite a long time. And then a colleague of mine noticed that in the species she was studying, it wasn't quite as mutualistic as that. Every time the female flew off, the male followed her. And so she changed the metaphor to mate guarding. And you know, when you do that, it changes the questions you have about the behavior. It changes the way you look at the behavior. It can even alter the kinds of experiments that you conduct on the behavior. And of course, there's also lots of unwarranted assumptions that come through anthropomorphism, assuming that animals are living in the same sensory worlds, living in the same worlds that we are. And of course, that's generally not true. Most animals have sensory systems that are different from ours. They see in a different region of the spectrum, or they hear higher sounds, or sometimes they even have whole sensory systems like electrolocation or magnetic location that are different from ours. So we can't assume when we see an animal do something, we can't just completely assume that we know what the animal was responding to. And of course, we always are assuming that animals have the same emotions that we do, and that they have feelings and so forth that are similar to ours. this is kind of a controversial point. I have no doubt that animals have feelings. I'm just dubious that they have the same feelings that we do. They might be, but I think it's a little dangerous to assume that they do. And this kind of anthropomorphism really colors the way in which you conduct your research or observe animals or think about the animals. Well, I haven't been so bothered by those kinds of issues because I've been studying what I've always kind of regarded as a Martian. It is just so completely different from anything else on the planet, and that is horseshoe crabs. And horseshoe crabs, they are arthropods. That is, they're in that great big group that includes insects and crustaceans, but they are not crabs. They are not crustaceans. They are in their own lineage that arose in the Paleozoic 500 million years ago. And there's nothing else that has before or since that's been like horseshoe crabs. They are most closely related to spiders, but they haven't shared a common ancestor with a spider since the Paleozoic, for 500 million years. So they are really different, and I could always kind of think of them as being different. They're also, our current day horseshoe crabs are also kind of like the horseshoe, are actually remarkably similar to the horseshoe crabs from the Jurassic. I don't think that's necessary because I've got that same picture here from the Jurassic. And this is an impression, or if you're on Zoom on the left hand, well, whatever, on one side there. This is the, one side has the oldest fossil horseshoe crab, and the other side has a Mesozoic Jurassic horseshoe crab. And this is an impression that was left in the mud on a inland sea of a horseshoe crab from that period. And I think that you'll see that they're actually very similar. It's just almost identical to our current day horseshoe crabs. So at least externally, there's been very little change in horseshoe crabs over 250 million years. So yeah, so in those senses, they are really regarded as living fossils. I got started studying horseshoe crabs many years ago when I first came to Florida because I wanted to take my undergraduate students out to the field station, out doing field work, get them away from campus, football games, all that stuff. And we had at Seahorse Key, it's 3 miles from Cedar Key, and it has a bunk beds and kitchen and so forth. So you could take classes out there. And so in about 78, I guess it was, I started taking my students out there. And the horseshoe crabs come to this lovely beach on the island, on the south side of the island. And the reason I wanted to take my students out there was I wanted a project that we could all do together. We could collect meaningful data and actually analyze all those data together. And this horseshoe crab really provided that opportunity because they're very predictable. They come to the beaches on the high tide, and in particular, they come to beaches during what are called king tides, that is the extra high tides associated with the new and full moon. And they nest along this beach, and in fact, they nest in a very narrow strip, just about a meter or two wide along this beach. And it's right at the very top of the high tide line, of the king tide line that they're nesting. So it was very predictable. I could take the students out and get lots of data, which we then analyzed together. Well, our first task, when I actually started this, there was really very little work that had been done on the individual behavior of horseshoe crabs. And in fact, they didn't know some of the most obvious things about their nesting behavior, their mating behavior. So this is really fun, just being able to go out there and start out anew with a Martian, with a very different critter. So the basic scheme is this. There's a female in front. She's the larger of the two. The male is in the back. this pair comes up onto the beach, and there's the male has some specially modified claws, front claws, and he hangs on to the back of the female. So he hangs on right to these spines here in the very back of the female. And as you see in the bottom picture there. And what he is doing is he is, as the female is laying eggs. I need a little anatomy here. The female is laying eggs. So here's what you see when you look down on top. There's this hinge here, and that allows them to dig, to burrow in and dig into the sand. And so there's this hinge on the top. And on the underside, there is a flap that's in front of the gills. They have these book gills that are actually rather similar to spider book gills. And underneath that flap is a, that here, underneath that flap that's right in front of the gills are what are called gonopores. They are the place where the eggs and sperm come out. So, so when you're looking down from above, the eggs and sperm are coming out right underneath this hinge. I'd also like to point out just one little other thing, which is that the mouth is between the leg bases. No other arthropod has this, current, regular, today. They chew food right here from the underneath side. Most arthropods have their mouth up front, but these have it down between the leg bases, like trilobites and eurypterids and stuff. And there was one other, oh yes. And so the animals breathe by water coming in through this area right here and coming back across the gills on the underneath side. So so they're circulating water across their gills as they're nesting or as they're doing anything else. So what is happening then is that here we have a pair nesting, the female's in front, the male behind. We put in flags every time the female stops. And when she stops, she lays her eggs and kind of pushes them down into the sand. And as she's laying these eggs from the gonopore that's right underneath that hinge, she's laying them through a cloud of sperm that the male has set up, ejaculating underneath. So this is a mating system. This is a reproductive system that no other arthropod has. The males are actually putting out sperm that is free swimming sperm underneath the female into the environment. It's a really peculiar system. So we put in these flags, and that then allows us to, wherever they stop, and lay eggs. And then that allows us to come back afterwards and dig up those eggs. And then we can learn a lot about depth of the nest and how many eggs are laid. Something like 60,000 eggs are laid, not at one time, but in little batches, as you saw, little batches of about 2,000 each. The eggs develop about that far down in the sand. The eggs develop, and they are actually developing because they're at the top of the high tide line. They're developing above where most tides will come in. And so they develop in wet sand for about 2 weeks. And then when you get another extra high king tide, then the water then comes in across the larvae again, and they then emerge from the sand. So 2 weeks later, they're ready to emerge, and they come out, swim out, and then they'll settle in the area just off the breeding beach. Did I get it all? Yeah, okay. And so we learned by tagging the animals, by giving them individual tags, which it's a very common thing to do in animal behavior, is to tag the individual, watch the individual behavior. We learned that they are producing about 2,000 eggs in each of these nests. And then the females nest for a couple of days. They'll come on the extra high tides for 2 or 3 days, and then they're done. They've laid all their eggs for the year, and they go back out to sea. The male lets go, and goes and hunts down another female. The males then are returning repeatedly. Sometimes they find a female, sometimes they don't. So males are returning repeatedly to the beach. And that means that there are a lot more males on the beach than there are on the breeding beach than there are females. And this sets up an interesting situation. So here we have a female that is nesting just by herself right here. The female is in front buried, and the male is the attached male behind her. Remember, they attached by those claws. There's a female coming in, and then there's 2 males that are unattached that are also approaching the breeding beach. And then in this picture, the female is nesting up there, and the attached male is back here. And 2 of those unattached males are gathering around this spawning pair. What appears to be going on then is that there are satellite males, as we chose to call them. There's other names that have been used, but I tried to pick a kind of neutral term. I didn't know what they were doing at the time. And there are satellite males and attached males on many of the nesting pairs. Okay, and so one of our first questions was the proximate question of how do those unattached males find the spawning pairs? Because it's actually more difficult than you might guess, because they're actually out of the water, and they're often up on the beach, and the unattached males cruise up and down just, in water about that deep off the shore. And horseshoe crabs are known to have very good vision. They have compound eyes. They are as sensitive. They see as well in the daytime as they see at night. So they have very sort of specialized eyes. And it was known already that they use their eyes to find females when they're attaching out in the field. Yes, they can walk on the beach. Not real fast, but they do walk on the beach. Yep. Yeah, so this male is like that top picture. He kind of identifies a pair and then kind of walks up onto the beach. Now, these look like nice pairs lined up here, but they're not. They are made out of cement. And my poor graduate student carted these incredibly heavy models up and down the beach. And she set up an experiment in the field with a pair. In this case, it was 2 pairs that were there. One was just a little bit larger than the other one. And these males that were working the shoreline there, these unattached males, find those pairs and were attracted to them, and on average were more likely to approach the large female than the small one, which makes sense because large females lay more eggs than small females. All right. We later attached some eggs and did some other things, and later learned that the males are also paying attention to chemical cues that are coming from the female and the eggs. It's another set of experiments. So that was the first step, the sort of proximate question. But then the ultimate question is, what are they doing? Why are they gathering around the pairs? And of course, I'm sure everybody has an idea. But one possibility is that these incoming males actually take over the attached male's position. And that does happen occasionally. They kind of come in underneath, but those attached males hang on for dear life. They are really hard to get off. And the female often leaves. If there's a fight like this going on, the females will often leave rather than continue with this. So takeovers are actually rare. Maybe 1 in 500 pairs that you see will there be a takeover. And so we thought it more likely that what was going on was that there was sperm competition between the attached male and the sperm that he's spewing out over the eggs and the satellite male. And the satellite males have a kind of favorite position that they go into. So here's a video of this. The female in front, the male behind, and there's a satellite just to the side. And as you see, they are focusing in on this region. You'll see it in this other guy who comes in. He focuses in right on, and he didn't fight. He's just comes in, goes right to that position over the incurrent canal. This is the canal that the female is breathing through. So she's bringing water in there and bringing it back across her gills. And of course, that also brings sperm if he's ejaculating. So, which he is. They're constantly doing it. And so you can see that this satellite just comes in, barges in, and then sets himself up at and you can get really big groups from time to time. So what we did was to watch these groups, and then we took tissue samples from each participant, from the male, the attached male, the female, and the satellites. We dug up the eggs and reared up the offspring. And then from those, we took DNA samples. And this is just like paternity analysis that you do on anything, on humans. And we compared the DNA from the female, the attached male, and the satellites. And the result of that was that the satellite males are doing very well, particularly when they're in that one specific position. They are fertilizing a lot of eggs. This actually surprised us that they were doing as well as they were. Even males in some of these, so this is the males in this favored, what I call 1F position, that right over the female's incurrent canal. They're doing just as well as the attached male on average. And the satellites, even satellites that are quite peripheral, are fertilizing at least a few eggs. So it actually then pays them, even though they aren't attached, to nonetheless come in, and even if they're number 3 or 4, to get in on the game. So what this means is that there are really 2 ways for males to fertilize eggs to gain reproductive success. On the one hand, they can go out to sea, find a female, attach to her, and come in when she chooses to come in. And on the other hand, they can come in without a female. The alternative tactic is to come in without a female and take your chances finding a couple and getting yourself into a good position. So there are what we call male alternative reproductive tactics in this species. These are found in quite a range of species. They're actually fairly common in the animal world, that there's more than one way of producing offspring. For example, in bluegill sunfish, there are parental males that guard nests and take care of offspring, but then there are sneaker males, as they're called, that come in, and they're a female mimic. They look just like the female. And they come in and spawn at the same time that the paternal male is spawning. And then there are also things like calling and non-calling frogs. There's quite an array of species that do this, that have alternative mating tactics. And in the calling and non-calling, one is calling and the other one sneaks in and mates with the female that is attracted by the calling male. And so these alternative, so as I said, they're very common, but they're nearly always based on size. It's a good generality that the primary tactic is the larger of the two. And usually the primary does considerably better than the secondary tactic. So these are alternative reproductive tactics are usually based on size. And I got all my students out there and we measured and we measured and we measured. And the attached and unattached males are not different in size. There are a lot of things I don't know about horseshoe crabs, but that I know. They are not different in size. So that really surprised me, needless to say. I was expecting that they would be. And that really surprised me. And then one of my students pointed out that yes, they're not different in size, but they look different. And you can see it in this picture right here. This is the female, the attached male, and the satellites. And the attached males are not different in size, but they are lighter in color, as you can see right here. On average, they are lighter in color. Now, there's something I haven't told you about horseshoe crabs yet, and that is that they take 9 or 10 years to develop, and they go through a whole series of molts. They go through 16 or 17 molts, and I have some examples up here of some of the molts that they go through. They start out, just tiny little horseshoe crabs, and then they each year they will molt and develop into something like this. At the time that they mature and become reproductive, they stop molting, and at that point, then they continue to live for maybe another 6 to 8 years, and during that time, their shell deteriorates. So they accumulate algae and blue-green algae and things growing on them, and the outside shell gets kind of worn off. And so you can see very clear differences between animals that have recently molted into the mature animal, and older animals that have not. So what this means is that the attached males have on average molted more recently than the satellite males. So attached males are lighter in color, they have less fouling on them, fewer things growing on them, and they are younger as adults than are the satellite males, because they have this terminal molt. And there's a lot of differences between younger and older animals. The younger animals are slower moving. The younger animals are faster moving. The older animals are slower moving, of course. And they will pair up occasionally. These, particularly the medium animals, we usually separate them into young, medium, and old. And the medium animals will pair, but they take longer to pair. We put little wading pools along the shoreline and allowed them opportunity to pair up. And yeah, the young animals paired very quickly in those pools, and the older animals were much slower to do that. And then when you went to release them as a pair that had formed in the wading pool, the older animals were more likely to let go. So there's just a host of differences between young and old animals. So that led us to in science, there's always, every question leads to another question. This is just the way it is. And we wondered why it was that the older males don't bother to attach. And we were doing some experiments where we were holding animals in little holding tanks. And I noticed that these attached males, the younger ones, the ones that had come in with a female, did not poop in those little tanks, which means they weren't feeding. And in fact, you can sort of understand why that might be, because the males are holding on right here, and their mouth is right under here. So the female's tail is right where their mouth is. And although they can pick up a few things with their claws, they're not feeding the way satellites and the way unattached males and females are feeding. So we did stomach analysis and fecal analysis, and then ran an experiment in which we had the animals in these little holding tanks and didn't feed them for a couple of weeks, and we showed that those animals were actually starving. That is, they were using their own tissue for metabolism. And so since males remain attached, in fact, we think that they may pair up earlier in the spring. The attached males pair up fairly early on, and so they may be attached for some weeks. And so our sort of current working hypothesis is that these older males simply cannot afford to go that long without feeding. So this is one of our findings. Well, I showed you this picture a little earlier as a way of showing you that there were two mating tactics for males. But you know, this picture shows something else that's kind of strange. And that is that when you walk on the beach, some of the females are attracting satellites and some of them are not. Females that attract satellites will have multiple male paternity. They'll have their offspring will be fertilized by, their eggs will be fertilized by the satellites as well as the attached male. So they'll have several fathers for their offspring, whereas the female that is nesting alone has only one father for her offspring. And so there really appear to be two ways for females to nest as well. And we learned fairly early on that these pairs were producing these pairs. We call them polyandrous, so I'll just call them polyfemales. The polyfemales were actually producing chemicals that attract the satellites, whereas these females are not producing those chemicals. We learned that by removing the satellites from a poly pair, and so you walk along the beach, you remove the satellites, throw them back in, and then and then you come along 10 minutes later, and the females that had satellites before now again have satellites, different ones, they all again have satellites, whereas those mono females still have not attracted any satellites. So it really does seem to be two ways for females to be nesting as well. So we decided to investigate this problem, and it's a kind of simple-minded hypothesis that if they are not attracting satellites, satellites must be costly for the mono females, and satellites must be beneficial for the polyfemales. And we measured reproductive success just in the sort of immediate sense of how long they spent on the beach and how many eggs they laid. So we ran these two experiments. In one experiment, we added satellites to these females, in which case we expect them to do poorly when the satellites are added. And over here, we removed satellites, and we expected them to do poorly there in this experiment. So here's an experiment. Here is the satellites added experiment with a mono female nesting here And on the beach, we moved the camera. And we've got our flags in so we can dig up the eggs. Now, they don't really like to come in. They're not interested in coming in. But if you guide them in with your foot, you just kind of urge them in, they will come in. And they interact with the attached male there, you see. And look, the female is already bending. And the two of them are interacting. And as soon as he's sort of released, she's able to get out. Off she goes. She does not like satellites. Those mono females do not like satellites. And so when we measured the reproductive success by measuring the number of eggs laid, how long they spent on the beach, they left very quickly. And of course, they laid very fewer eggs than if they were just then the controls, which were just allowed, they were mono females that were allowed to nest by themselves. So it was very costly for those females, for the mono females, to have a satellite. The other experiment was one in which we took the satellites away from a polyfemale. And it's hard to do, actually, because the satellites are so attracted to them. And you have to kind of guard the pair that you're watching to make sure that nobody comes in, no satellites come in, and they're constantly trying to get in around your flags and so forth. But the results of this experiment were very surprising. So these are polyfemales, and we are keeping the satellites away, as compared with the control, which is polyfemales, where we allowed the males to stay there. And polyfemales without males did much better. They stayed on the beach longer, and they laid a lot more eggs than the females that had all those satellites. Big surprise. Did not expect that. And so we figured, I mean, this is a hard experiment. The next level is hard to do, but we figured it must be that the polyfemales are getting something from all these males, and it must have something to do with the development of the eggs. And so our hypothesis is that the polyfemales are mating multiply, attracting in all these satellite males, and that this improves the development of their offspring. So we did in vitro fertilization, which is done just like it would be done anywhere else. We took eggs from a female and distributed the eggs into a number of different containers. And then for half of those containers, we put in the sperm from her attached male, that female's attached male. And then in the other half of those containers with her eggs, we put in the sperm from a satellite male. And then we did the same thing with mono females. We had mono females' eggs, and we compared the success of her own attached male with that of a satellite male. And the results, again, well, in one sense, they were great. In another sense, this was a little surprising. The mono females did just as well with the satellites and the attached males. The proportion of eggs that developed under these in vitro fertilization conditions, which aren't perfect, but this is the proportion of eggs that develop, were the same for monandrous females, for satellites and attached males. But for polyfemales, they did worse with their own attached males. They had fewer developing offspring with their own attached male sperm than with the sperm from a satellite. They did better with the satellites. So our conclusion here was that there is something wrong with the males of the polyandrous females. And of course, it has many possibilities. But one of my students was doing a study on sperm counts in horseshoe crabs, and for another reason. And so we got him to look at the sperm counts for the attached males of monandrous females and the attached males of polyandrous females. And from that, you can see that the attached males of polyandrous females have lower sperm counts than the attached males of the monofemales. So this is at least suggests one possibility of what might be going on is that these females, these polyfemales, are attracting satellites, are producing a chemical, attracting satellites to join the nesting when their own attached male is in poor condition. In fact, the data are that the younger males, and on average these are younger males have higher sperm counts. And so if a female finds herself with with a male who has a low sperm count, male in poor condition, then she produces a chemical that attracts in the satellites. So that's our current working idea, our hypothesis. Yeah, it's something that we didn't actually expect at all when we first started working on this. So I hope that you've been able to get a little bit of a feeling for how we do research in animal behavior. I think you'll find some of the others people do research a little differently than I, but it involves a lot of observation, description, identifying the behavior of individual animals, looking at what they're doing, asking both proximate and ultimate questions, often going back and forth between proximate and ultimate, and then doing experiments. And many of the experiments that I've done have actually been field experiments, which we tend to think of experiments as being run in the lab and field work as being observational. But you can do perfectly good experiments in the field and find out, really what the animals are doing under natural conditions. Yeah, sort of all along learning about the life and the world of this very bizarre animal. So I'll stop right there and take any questions that people might have. So there's a question right here.
Do I have any volunteers to help me with the microphone?
I could.
Thanks. Did you say that they lay as many as 80,000, or is that in a lifetime?
Nope, that's in a year. So they lay them in batches of 2,000 or 3,000, 2 to 4,000. And, where I put in my flags, there's a couple thousand each. And if they stay there long enough, they can lay all of their eggs in all the eggs they have in one afternoon or a couple of afternoons, a couple of high tides, and then they go back out to sea. But
so what is the rate of survival of all these eggs?
well, on average, if the population is stable, on average, it's 2. Right? I mean, if the population remains the same, then it has to be 2. But in our lifetime, yeah, there's a lot of loss. Yeah, and in fact, in some places, horseshoe crabs are not doing very well. And in New York area, there's the numbers are way down. Connecticut, Long Island Sound, the numbers are way down. Delaware Bay is being managed, and they're doing much better there. The numbers are fairly stable. Here in Florida, they've never been studied until we started working on it. We've started a citizen science program to survey and tag animals. So we're now just now learning whether the populations are stable or not in Florida. Some populations look pretty good, others not so much, yeah. But really, we haven't had our 10 years. You know, when an animal that takes 10 years to develop, you know, you have to go a long time before you can get a good measure of the population stability.
If a mono female gets an underperforming male, can she become a poly?
I don't know that. It's possible. The mono females do tend to be, on average, a little bit younger than the poly females, you know, when we really looked at the whole data set. But you know, most of them are medium or in between. So my guess is that they can. But how do they know? How do they know the condition of their
question.
male?
What makes a mono versus a poly?
Yeah, exactly. How do they know? That's one of the things that made it such a surprising result to us, because somehow they have to detect the quality of that male. I don't know how they do that.
Jane, you mentioned that they are eating while they're burrowed in, the females anyway.
Oh, not
And-
during nesting. Not during
okay.
nesting.
I was
No.
wondering, what do they eat?
So when they go back out to sea, by that I just mean offshore, they're usually found within 30 feet or so in shallow. The ones in the Cedar Key area never leave that big bay area there. They feed on clams, and given the clam industry in Cedar Key, but the clams there are in bags, so it's okay. So they feed on clams very heavily, and they feed on worms and other invertebrates that are in the sand. Yeah.
They're sort of conspicuous, and I wondered what eats them.
Oh yeah, there's a lot of things eating them. We had a graduate student in biology that found 17 horseshoe crab parts, the parts from 17 horseshoe crabs in the stomachs that he was analyzing of alligators in the Indian River. So alligators, the juveniles are eaten the eggs are eaten intensively by birds. And in fact, there are some species of birds that depend on horseshoe crab eggs. They actually time their migration to that. The juveniles are eaten by birds, anything else, crabs, anything else that can. And the adults are taken by rays and alligators and, yeah, probably lots of other things as well.
We have hands flying everywhere, but
I have the microphone. I got it. All right. A question about the use of their blood. I remember reading research where they're using blood for, for health reasons.
Yep.
Can you address that?
Sure. The horseshoe crab blood, this was discovered completely by chance. Horseshoe crab blood has in it a chemical that is extremely sensitive to gram-negative bacteria. And after this was discovered, they actually developed a test called the LAL test. Limulus is the genus. Amoebocytes, they're little cells in the blood that are broken open, and the chemistry inside is sensitive. And there are 5 companies that bleed horseshoe crabs. They take about a third or half of their blood and spin it down and break open the cells and use that chemistry in what is called the LAL test. And the LAL test is used for anything that is put in the human body, anything. You know, hip replacements, knees, heart valves, IV fluid, vaccines, anything. It's the standard for anything that goes in the human body, or veterinary medicine also. Hundreds of thousands are taken, are bled. There's a little bit of disagreement about how many die in that process. It's probably 15 to 30% will die as a result of having so much of their blood taken from them, and also just being taken out of the water and carted around, and put back again a couple days later. It is regulated, and they are trying to be as nice as they can, but it's, however, as of 1st of September, there has been, this happened many years ago, they cloned the gene for this substance, this chemical, and they cloned the gene and put it in yeast, and it's called factor C. The yeast now produce this same substance, but it was never accepted by the pharmacopoeia, you know, the thing that's handing and taking care of all the drugs. It's determining the conditions under which drugs are kept and are manufactured. And so it hadn't been accepted, but the pharmacopoeia has just now put this out for the factor C out for being considered. And so maybe in a year or so, those folks will be out of business.
All right, we have Roxanne, and then we have a
Yes.
couple chat questions. Let's do
Okay.
Roxanne. Go ahead, Roxanne.
Oh, I wanted to ask, are horseshoe crabs found anywhere? But the East Coast, are they found on the West Coast or other countries?
Right, okay. So Limulus polyphemus, which is our American horseshoe crab, is found from Maine all the way down around Florida over to Mobile Bay, and then it's also found on the Yucatan Peninsula. Funny distribution, but that's the way it is. Then there are 3 other species of horseshoe crabs. They're different genera, so they're fairly distantly related, but they are horseshoe crabs. And they are found in Japan, along the coast of China, Myanmar, Vietnam, Myanmar, and up into the Bay of Bengal. So there are 3 species in that area. They are doing very badly. The Japanese one is probably close to extinct. And they, by the way, have also been bled for for TAL rather than LAL. They make their own. The Chinese are making their own from their own horseshoe crabs, and so this is probably going to make a huge difference for those animals, those populations.
Okay, can you see the chat questions, Jane, or would you like me to read them?
Do the females change from monandrous, polyandrous over their lifetimes, or is it only based on the attachment? Well, we don't totally know that, but they do. They can change. Some females can go from one to the other. Yeah, it's a little bit hard to tell because on a day, some days there's a lot of satellites out there, and some days there aren't. And so sometimes we probably misidentify some of the females, call them mono when they're actually would prefer to be poly, that kind of thing. So it's a little bit hard to be sure about that, but I think they probably can. And as I mentioned, the maybe I didn't. The monandrous females are on average slightly younger, so it's might be something that's partially something that changes with age.
And then the second
Jane?
one was, what is the role of horseshoe crabs in the marine ecosystem?
Ah, yes. Well, I got started on that. So the eggs are eaten by lots of shorebirds, and as I said, red knot, this one species that's actually endangered, depends on the red knot, depends on horseshoe crab eggs, times its migration into Delaware Bay so that it can just eat. There's an unbelievable number of horseshoe crabs in Delaware Bay. It's just, I didn't show a picture of it, but oh, it's just ridiculous. They were just piled on top of each other. And that's on purpose. They're being managed, and it's on purpose because horseshoe crabs will, when they're digging into the beach, they will accidentally dig into the nests of a previously nesting female. And that then results in the eggs coming to the surface. And that's where the shorebirds can now feed on the eggs. So they are being maintained at a sort of artificially high level in the Delaware Bay.
Jane, back here.
Oh, hi.
Hi. Thanks for the interesting talk. I'm just very curious, from the force of the waves coming in over them while they're mating, Doesn't the sperm wash out?
Yeah, I don't know. I don't understand the whole system. I've worried about that too. And they are sometimes even out of the water, but they're in a kind of slurry of sand, and you go down a little ways and it'll be a slurry of sand and water, and so I'm not sure. I have some evidence from another issue that the females actually hold sperm underneath the shell, so that if they're going through a series of nests, well, I can explain. If they're going through a series of nests of those 2,000 eggs each, if they had a satellite back here at the beginning and he left or we took him away or something, some of the eggs fertilized up here might come from him. And so it looks as though the female is able to somehow, they're kind of concave, particularly up in here. And so it looks as though the females are somehow able to hold sperm underneath their shell.
I assume pheromones play a role in the attracting of males to females. Do the mono and the poly females have the same pheromones?
Yeah, we don't know anything. We know that they have them. I've just called them chemicals because I don't know exactly whether they could be just odors coming off the eggs, or, it could, I don't know, it could be other things. But my suspicion is that it is a pheromone. It stays in the sand after the female has left, and satellites will sometimes come up and are really attracted to the area where a female. So that gives me a kind of a clue that there is a real chemical in that sand. Gosh, I've lost the question.
About the pheromones attracting the males to the female.
Yeah, I think they are
and the
pheromones.
different females have the same pheromone.
Yeah, no,
You don't know that yet.
we don't really know that.
Yeah.
We don't even know what pheromone is, right?
My question is more general. On a species like this, sea animals that have gills, but how long and how do they live on land? Weeks and weeks.
Yeah. Well, they're not on. Okay, so when they come up on the beach, and they're nesting on the beach, and their gills are out of the water, they cannot breathe. But the horseshoe crabs are very tough. It's hard to kill the horseshoe crab, and so they actually just go into anaerobic respiration while they're on the beach up there. And that same thing is true when they are brought in for bleeding by these companies. They're not breathing during that couple of days, but that doesn't kill them. They're able to. Oh, you've probably seen them on the beach upside down. They can stay that way for a couple of days if they don't get too hot. They can stay that way for a couple of days. Now, the juvenile, so the eggs are laid in the sand, and the eggs have a little embryonic membrane around them. And there's enough oxygen in the sand, and the sand remains wet during that 2 weeks of development. And so the embryos live quite well because there's plenty of oxygen in the sand, and they're protected. They actually swim around in their little embryonic membranes, and they have water there that has exchange just like an egg. But when the tides come in, that causes the embryonic, when the king tide comes in again after 2 weeks, that causes that embryonic membrane to break open, and they then swim out. Those animals they cannot stay on the beach. They will die if they are out of water. So they're not really out of water when they're developing. No, I think the only thing I remember about 1 or 2 weeks was, I see. No, I'm sorry. They come up just for the high tide, at the time of the high tide, and then they go back out after the tide has receded. And then they will come back in on the next high tide, do the same thing again, and they'll do that for a couple of days on those extra high tides, and then they're gone for the season. I'm sorry, I probably neglected to say that they leave when the tide recedes. Yeah. Yeah, Ray.
Has the red tide and climate change affected it? And then also an add-on question. Since you're a beekeeper, do you see any relationship between crabs and bees?
The red tides don't seem to bother them too much. Occasionally there are some die-offs of horseshoe crabs in Indian River, but they don't really seem to be, it doesn't seem to, that's associated with red tide. However, climate change is definitely affecting them. I have seen this just since I've started studying them. They're beginning to nest much earlier in the spring. They didn't used to begin to nest until March, but now if the water warms up, they'll be nesting in February. And the same thing is true going up the coast. So they're nesting earlier in Delaware Bay, for example. Well, that's a problem because those birds have timed their breeding to the horseshoe crabs. And if the horseshoe crabs are breeding early because the water's warm, then they're going to miss. There's going to be a miss. Beekeeping, I haven't thought about that too much, except that we use the same method, same observations. I mean, there's a lot of animal behavior in beekeeping, that's for sure. But I don't know very much about the mating system. I mean, I do know what they do, but I don't know anything about the details of the mating system. So I've got more to learn about those honeybees. Yeah.
Jane, I know you said that when they get to maturity, they're not molting anymore. And I don't know how many gazillion of them you have there. Is it possible to tag them and see them again the next year to see if the behavior has changed, if they're mono 1 year and poly another?
Yes, yeah, we do. We have tagged them, and we're tagging them also for this citizen science project. Yes, we have tagged them. However, there's a lot of them out there, and our returns are such that you can't really get enough data to be able to say certain things like that. I mean, we know that there's satellites, attached males. Males are sometimes attached and sometimes satellite, and there's some patterns to that. In other words, an old male is nearly always, right. Yeah, yeah, we should be able to, but it just, life is short, right? Yeah. Since they are related to spiders, how did they get that hard shell? Yeah. Well, spiders have hard shells too. The shell is actually made almost entirely of chitin, and which is in the shells of other arthropods, but they don't have calcium in their shell. And so they don't have the problem of hardening up the way a lot of crabs do. But that's also what makes it hard for them to make good fossils, because they're not, it's not hardened material the way it is for crabs and, snails and things like that.
Jane, thank you for sharing your passion with us, and you just definitely kicked off your course.
Good,
Appreciate
yes.
everything you've done.
Oh, I hope that you will come next week. Looking forward to hearing from my colleague, Nicole Gerlach, who is going to tell you about another mating system, which also seems to have a complicated sex life, so-called monogamous.